Good morning, everyone. Welcome to today's webinar. Thank you for taking out your busy schedule to be part of this webinar. My name is Yiling, and I will be the moderator for today's session. Our topic today is mRNA vaccine in a lab from plasmid to mRNA. During the webinar, feel free to ask any question in the chat. We'll do our best to address them in the Q&A session at the end. Now, without further delay, I will hand it over to our speaker, Mr. Dong Chi Ho.
Hi, thank you, Yiling, and good morning to everyone. So I will actually upload my slides, so give me a moment. Okay, Yiling, can you let me know whether you can see my slide?
Yeah, can see your slide.
Okay. Hi, good morning, everyone. Thank you for joining the session today. Today, my name is Ji Hong. I'm from NextGen Scientific. Today, I'll be covering a little bit on the topic mRNA vaccine in the lab from plasmid to mRNA. So as a summary, today we are going to talk about the design of the template, which normally is the plasmid construct. Okay, what should we actually input into the plasmid? And secondly, we are actually looking into some essential elements of mRNA, what option we have for this element, and finally, about the in vitro expression of this plasmid to become mRNA, what kind of option we have. So I will begin today's session with some background and introduction to mRNA vaccine. So mRNA vaccine technology is based on a vehicle that enables the delivery of a nucleic acid molecule encoding the antigen of interest into the target cell in the host, which is the human. By doing this, is allow the host cell to express the antigen to elicit the immune response. So normally these mRNA are encapsulated in a delivery vehicle such as lipid nanoparticles, short form the LNP, which allows the entry of the target cell through the process called endocytosis. So by going into the cell, it will actually form an endosome. So normally endosome fusions will be the entry of the endosome formation will be actually fused with a lysosome, and the lysosome contains enzyme that degrade whatever is in the endosome. So therefore, for this part, we need to prevent it from happening to evade and allow the expression of the antigen protein. So now we should move on to the mRNA structure for our mRNA vaccine. So the structure of an mRNA vaccine is very similar to mRNA in the native state. So over here, on top here, we have the structure of the mRNA. So the structure of the mRNA of the native molecule consists of a 5' cap, a transcription start site, and then a 5' UTR, start codon, the coding region, the stop codon, continue with the 3' UTR, poly A signal, which is actually the poly A tails. So this is the native structure of an mRNA, and we need to include the cap, the untranslated region, coding, and poly A tail, so that it forms a complete molecule, which is actually the mRNA. So in order to produce this particular molecule in a single-stranded, first we need to construct a template. Normally we are using the plasmid strategy. So over here you can see that we have an additional element, which is actually the promoter controlling the transcription of the mRNA. Then we continue with the UTR, the untranslated region, coding, and so on and so forth. In addition, normally we will actually include 2 restriction endonuclease cutting sites flanking the front and back of this particular design as well. Okay? So that after we got this construct, we will actually digest to release the double-stranded template. So again, by having this double-stranded template, we can then move on into the in vitro transcription of the mRNA. So basically, normally we'll use a commercial kit that contains the T7 RNA polymerase or any RNA polymerase that can recognize the promoter that we use in our template. So the RNA polymerase in the kit together with the nucleotides will be producing the new mRNA transcript, okay, with the structure that we designed. So finally, we clean up this particular mRNA transcript and then engulf it into a lipid nanoparticle and deliver it into our site for the production of the protein, and thereby trigger the immune response and produce the antibody as the effect that we want. So next we'll look at why mRNA vaccine is gaining so much popularity and become so important. So basically, number one, this platform exhibits several notable strengths over other methods. So the first one is it actually can acquire the native post-translational modification, and the conformation of the product, which is actually the protein, are very similar to the protein expressed from the natural infection. So therefore, it's promoting appropriate humoral and cytotoxic T cell responses. Secondly, the mRNA is non-infectious, non-integrating, and degraded in several days to a few weeks. Thus, there is no potential risk of infection, insertional mutagenesis, or uncontrolled protein production. So this particular point is actually relative to the alternative delivery method, okay, which is actually the viral method. Okay. So in the viral method, we actually include the mRNA sequence into a viral vector, and then we form a viral particle and we deliver the viruses particle and infect the targeted site to produce the protein. Third, as there is no anti-vector immunity against the mRNA, the mRNA LNP approach vaccine can be administered repeatedly. A notable impediment to the use of viral vector that I described in the second point. So as a result, the clinical success of nucleoside-modified mRNA LNP vaccines surpass other competing vaccine platforms due to their faster development, better safety, and highest level of protective efficacy. So now we move on to the first part of today's presentation, which is how can we construct the template for the production of the mRNA. So the first thing is we will need a promoter to control the expression of the mRNA. So there are several choices for the promoter. So the most popular one will be the T7 promoter, and then we have the option like SP6 promoter, so on and so forth. So why T7 promoter is such a popular choice is because it's a viral promoter that is actually used for bacterial protein expression. It's a very common promoter to be that particular purpose, and therefore we understand it very well. So by using the T7 promoter, we will need the compatible RNA polymerase, which is the T7 RNA polymerase. So one note about the T7 RNA polymerase is that it does not produce the poly A tail that is required for the structure of the mRNA. So the gene synthesis service could be used to synthesize the entire sequence, starting from the T7 promoter, the untranslated region, the coding region, the untranslated region, as well as the poly A tail as well. Okay. So normally we will have the RE, the restriction enzyme recognition site, flanking the 5' end and the 3' of this particular design, so that we can actually cut it up and to use it for the in vitro transcription. So the promoter selection will decide which in vitro transcription kit to be used, okay, as well as the choice of tab that we will discuss in the later part of today's sharing. So moving apart from the promoter, now we will take a look at the 5' untranslated region. So the 5' untranslated region of the mRNA are important to control the gene expression as well, because this part acts as an entry point for the ribosome during translation. It can adopt, elaborate RNA secondary and tertiary structures to regulate translation initiation. So when we have a very stable secondary structure within this untranslated region, it can actually impede the ribosome scanning, while if you have a less thermostable structure of this untranslated region, will also affect translation if it's close enough to the 5' cap. So another thing we should take note is there should be some appropriate distance of this cap to the 5' heavy. Okay? So the GC content, and as well as we should include a cosec sequence at the 5' UTR region. Okay. So what 5' untranslated region should we use? What are the choices that we have? So the globin gene 5' UTR sequences are often used to ensure high translation efficiency. In particular, sequence of the human alpha and beta globin, and another choices will be the rapid beta globin UTR region. So they are shown and employed to improve the translation efficiency of our mRNA. So the 5' UTR region of the rapid beta globin is thought to mediate efficient cap-dependent translation, and it does not contain any secondary structure or sequence elements that may negatively affect translation. So aside from the human globin genes, so we have another choice will be the heat shock protein, the HSP70, and then as the option of the UTR. Okay. So last but not least, there's another option which is actually the TPL of the adenovirus. It can also is shown to enhance protein expression by enhancing translation initiation. So as a sum up of the above, so a lot of the natural 5' UTR for those genes that are constitutively expressed can be used to enhance the translation efficiency of the heterozygous mRNA. So for the coding part of the sequence, things to consider is that we might want to include a cosec sequence in front of the start codon. For the sequence of the coding, we might want to perform what we call a codon optimization, as well as the GC content of the coding sequence. So codon optimization basically means that we alter the nucleotide sequence without altering the amino acid sequence. So using this particular table as a reference, you can see that one particular amino acid can be coded by more than one codon. By using codon optimization, we will use the most common nucleotide sequence that can be recognized by our host, which is actually normally of the vaccine is the human. Next, we will move on to this 3' untranslated region and poly-A tail inclusion. So compared to the 5' UTR, the 3' UTR regulate the fate and utility of the mRNA, including degradation, translation, and localization, as it have distinct regulatory signals that bind to the effector, such as RNA binding proteins, microRNA, and other non-coding RNA. So one of the examples is that the AC-rich elements at the UTR are known to destabilize the mRNA. And then the next thing is we should include a poly-A tail sequence. So there are two options that we can select to add this poly-A tail. So the first thing is we include a stretch of more than 15 A's during the gene synthesis step directly. Or it can be added after the in vitro transcription step using E. coli A-tailing enzyme. So which 3' UTR to use? So there are actually less studies on this 3' UTR compared to the 5' UTR that are related to the desired protein expression. So regulatory sequences of human globin genes are normally used as the 3' UTR to ensure the high stability and translation activity of mRNA. Of the research of Orlandini, it showed that 3' UTR containing the mTRNR1, which is actually the 3' UTR, small segment of 3' UTR of the 12S ribosomal RNA, and the AES, which is actually the member of the family of transducing light enhancer, are able to promote the expression of the coding sequence. So in addition to these two choices, active research group is currently considering using some non-natural artificial sequences of the UTR. Okay. So after looking at some of the choices of the 5' UTR, the 3' UTR, and about the codon optimization of the coding genes and sequences, so now I would like to share with you guys the case study from a publication that compare the mRNA vaccine of the Moderna versus the BioNTech, and then to take a look at how they make their decision and consideration during their design of the construct. So why I'm using this tool, or the publication why is using this tool, mRNA vaccine as a comparison, is because both of them are mRNA vaccine. This is number one. Secondly, both of them actually using the same target of amino acid sequence, which is actually the spike protein of the SARS-CoV-2 virus. However, although their amino acid is the same, their nucleotide sequences, especially as the Wobbles position, are substantially different due to the different codon optimization strategy used. Okay. So the first part we'll look at is the choice of UTR of these two mRNA vaccine. So the Pfizer-BioNTech 5' UTR sequence is derived from the above mentioned hemoglobin alpha globin gene. Okay. So it's actually an efficient expressor, while the source of Moderna 5' UTR is actually unclear. So I actually did perform a blast using this particular sequence, and it actually didn't come up or come back with any result. So the Moderna 5' UTR contains a GC-rich tract just upstream of the cossack sequences, and this GC-rich tract and its secondary structure may reduce translation initiation efficiency and overall protein output. For the 3' UTR, the Pfizer-BioNTech vaccine combines one segment of the human mRNA encoding enhancer, the AES, and the second part of it fused with a mitochondrial 12S ribosomal RNA, the mTRNR1. So in comparison, the 3' UTR of the Moderna vaccine is actually using alpha globin HBM gene. So the reason why Pfizer-BioNTech actually choosing a fusion 3' UTR, the AES mTRNR1, was that because in their own testing, they show that this particular 3' UTR outperformed the beta-globin 3' UTR in terms of mRNA stability and protein output. In the publication of Zhang in 2023, they altered the 3' UTR of the Pfizer-BioNTech to become beta-globin, and their findings show that the fusion UTR AES mtRNA does not showing more efficient translation compared to the HbA1 3' UTR used in the Moderna vaccine. So the study also suggested that the effect of this UTR on mRNA stability could be as well dependent on the type of cell or tissue type as well. So for example, the lymphatic compartments versus the muscle with a tissue. Here is from the same publication, they're showing the 5' UTR sequences of the Pfizer-BioNTech, the 3' UTR sequences of the Pfizer-BioNTech, and Moderna 5' and 3' UTR. Okay. So as mentioned previously, the Pfizer-BioNTech choose to use the HbA1 5' UTR. Moderna is unclear. For 3' UTR for Pfizer-BioNTech is a fusion of AES and mRNA. For Moderna, it's actually a HbA1 gene. So next, we'll look at the codon optimization strategy of both mRNA vaccine. So we will do the comparison in terms of the GC content, as well as the use of pseudouridine. Okay. So as mentioned earlier, both Pfizer, BioNTech, and Moderna vaccine encode the identical spike protein of SARS-CoV-2. Okay. So, however, their nucleotide sequence are actually different, especially at the global place. Okay. So this is due to the codon optimization strategy that they use. So for Moderna, during their codon optimization, they replace most of the adenine and thymine at the global position with guanine and cytosine. So as the end result, it leads to a 14.5% higher GC content at the global position compared to the Pfizer-BioNTech. So although high GC content might be beneficial for mRNA translation or stabilization, it reduces the content of uridine in the mRNA that can be replaced by the M1 pseudouridine during the in vitro transcription. So the M1 pseudouridine enhances vaccine efficacy by stabilizing the mRNA and facilitating the evasion from the host innate immunity. So in a regular mRNA, higher GC content compared to higher GC content actually increasing mRNA stability. So it actually correlates with higher protein expression in mammalian cell. So in Pfizer-BioNTech, because they didn't have such a high GC content, they decided to change the uridine of the nucleoside with a modified nucleoside that is called M1 pseudouridine. So this particular methylated pseudouridine promotes the mRNA stability via increased base pairing and stacking. So based on the study of Zhang publication, they are unclear that whether which one is better by using the methylated pseudouridine for the protection and stability of the vaccine, or by codon optimizing all the A and T to become GC content. It's possible that both are able to work together and compensate each other on the mRNA stability and antibody production. So both of them might actually working together. Okay. So moving away from the comparison of the strategy used for the Pfizer and Moderna. So now we'll proceed to the next step of the mRNA vaccine. So after having the construct ready. So we'll move on into the in vitro transcription part of the mRNA vaccine, which is actually producing the mRNA in this particular structure. So with a 5' cap, as well as all the elements from the mRNA, and finally the poly-A tail. For the in vitro transcription to happen, normally we'll be using a commercial transcription kit. So the kit will actually provide the polymerase, the substrate, which is actually the NTP. Then we put in the template that we release from the vector, which is actually linearized. And the enzyme will recognize the promoter and produce the mRNA as the final product. Once we have this final product, we'll do a cleanup and we will actually package it. So what are the considerations that we should have when we actually choosing the in vitro transcription kit? So the first part of this consideration will be which cap that we want to use. So in eukaryotic cell, most RNA, mRNA capping enzyme will add a 7-methyl guanine 7-mg structure to the 5' end of a nascent pre-mRNA. So this modification is important because it prevents RNA degradation, it promotes translation, and modulates its immunogenicity. So research and development objectives are best achieved with careful selection of the RNA capping approach. So in the intricate world of mRNA vaccine, mRNA synthesis, the cap structure plays a pivotal role in controlling the gene expression. So scientists have made significant effort and development in giving us choices of various cap analog to enhance mRNA stability, translational efficiency, and therapeutic potential. So the cap choices that we have so far will be like the M7G cap, so that used in the native mRNA. Then we have the ARCA cap, as well as the clean cap. Okay. So this cap is again a modified guanosine nucleotide added to the 5' end of the mRNA during transcription. So the first choice that we have for the mRNA cap is the M7G cap, that very similar to the native mRNA. It features a methylated guanosine linked to the mRNA via a 5' to 5' triphosphate bridge. So from over here, you can see that it's actually a 5' to 5', and then allow the 3' end to have the extension of a new chain, which is actually a bit different from the structure of the extension of the base that we see in a polymerization reaction. So however, by having two OH group at the 3' end, the new extension can happen either way. But we want the cap to be the methylated G, but not the other guanosine. So therefore, this is the challenge of the M7G cap. Sometimes we call it the cap O. So the second choice of the cap will be the ARCA, the anti-reverse cap analog. So it's considered the solution to the cap O because we are adding a methyl group to the 3' end of the M7G. Okay? So this is the same M7G, and we add a CH methylation group over here and prevent the extension, the new chain happen over here, so that the new chain extension can only happen to this particular guanosine. So therefore, methylation group, a new strand extension. So the new mRNA will actually have the methylated cap. Okay? So, but ARCA cap has its own problem as well. It can only generate cap 0 structure, not cap 1. And GTP will be compete with this ARCA cap for the first position on the mRNA. So to solve the issue of the ARCA cap, the commercial company or the researcher group actually come up with a trinucleotide cap analog. So this one is actually having a feature of cap 1, meaning that the first nucleotide of the mRNA has a 2O methyl modification as well. So just now we mentioned that we add a methylation group here to prevent a new extension, which is actually the ARCA cap. So cap 1 actually having another methylation site, which is actually at this particular part of the nucleoside as well. So the extension actually happened in this third nucleotide. Okay. So if we compare closely, the ARCA cap only contains 2 nucleotides, whereas the cap 1 structure actually contains 3. So the first, the second, and the third. So this whole thing is actually a cap 1. So yeah, so we solved the issue by having a third nucleotide in the entire cap structure using cap 1 And this particular cap, sometimes we actually call it the clean cap. So by having an unmethylated G after the 2O methyl A, so the mRNA chain can now be extended from the third nucleotide. The new cap is actually initiated with a AG initiating mRNA transcription. It will not compete with GTP for chain extension. So capping efficiency and RVT in vitro transcription yield could be improved over the use of ACAR cap. So the company trialing first commercial this particular product under the name Clean Cap AG, and a variation of this molecule is called Clean Cap reagent AG with the methylation modification. And this particular this capping is actually the choice to manufacture the vaccine of Pfizer-BioNTech. Okay, so the 5' cap is, especially the cap 1 structure, is critical to facilitate translation, initiation, and protect it from degradation. Okay. So therefore, many RVT mRNA are using this particular approach. So after we choose this particular capping, we actually have to decide whether we want the capping to happen during the synthesis of the new chain or what we call a post-translational enzymatic reaction. So basically, it means that in the extension of the transcription event, the IVT reaction, we add in the cap, or we wait until the whole IVT reaction finish, then we actually adding the cap. So the 5' cap of the Moderna vaccine was added post-transcriptionally using a vaccinia virus. And although post-transcriptional enzymatic reaction can achieve 100% capping efficiency, the process is actually higher cost and take longer compared to core translational method. Okay, so in contrast, the clean cap will now provide nearly 100% capping efficiency in the Pfizer-BioNTech mRNA vaccine. Okay. So these are some of the structure of the clean cap offered by trialing, whether you have the methylation on the second nucleotide. So the addition of the methyl group on the position 8 over here are shown to have further increased protein expression. However, the real mechanism is remain unknown. It has been hypothesized that this modification actually positively influence the mRNA stability and prevent enzyme mediating the capping. Okay. So after choosing the cap and whether to add the cap during the in vitro transcription reaction or after it, so yeah, will be the process. Okay. So this is showing a process that we actually performing cap adding after the transcription. So we get hold of the mRNA first, then we add the capping enzyme, and then to get rid the final product. So the other choice will be what we call a core transcriptional, where the cap is added into the in vitro transcription reagent, and the final product will be the mRNA with cap. Okay. So core transcriptional kit nowadays has been improved a lot, and it can actually produce very high capping efficiency as well, not very much less than the post-translational method. Okay. So although post-translational methods still give the highest capping efficiency, okay, even on difficult substrate if you are using the Fausto virus capping enzyme. So this capping enzyme only responsible for the capping function. If you would like to have the methylation on the 6 site or the 2O site, you need another enzyme, which is actually the 2O methyl transferase. Okay, so in the same reaction of the post-translational capping, so overall, the core translational capping of the mRNA with the template encoded poly-A tail is recommended for most bench scale application, whereas enzymatic capping is the recommended approach for large-scale mRNA manufacturing. Okay, so this is another highlight if you would like to use the clean cap. So because the clean cap is being used, there will be a slight modification on this T7 promoter use. So on this particular, the last two base of the T7 promoter, if you would like to use the clean cap, it should be changed to the AG base. So another consideration during the in vitro transcription will be the use of pseudouridine. Because without the use of pseudouridine, the in vitro transcribed mRNA would be prone to nuclease degradation when injected into animal. So they will lead to some innate immunogenicity similar to what happened when infected by a pathogen. Okay. So the problem for the degradation will be the use of pseudouridine, which can be used to replace the uridine in the in vitro transcription reaction. So incorporation of this modified nucleoside actually found in natural RNA as well. So it reduced the TLR-mediated innate immunity against the mRNA and increased the translational capacity and biological stability of the mRNA. So a possible better choice of the pseudouridine will be the M1-methyl pseudouridine. So more recently, it's actually also found in 18S RNA, and it actually showed to outperform the pseudouridine in mRNA modification by offering better expression as well as reduced innate immunity. So considering the beneficial effect of this M1 methyl pseudouridine, okay, so both Pfizer-BioNTech and Moderna actually replaced all the uridines of their mRNA vaccine to become the N1 methyl pseudouridine. So after successfully getting the mRNA, so you will need to clean up it and then package it into the delivery. So for the delivery of the mRNA, normally you actually pair with the use of lipid nanoparticle. So the lipid nanoparticle delivery is actually very popular to be used for the mRNA vaccine. Okay, so it's actually a breakthrough research on this particular area because the development and optimization is very crucial. So LNP formulation are actually using the ionizable lipid, okay, and it has gained attention because they are positively charged at acidic pH, and this is have a crucial point because it can actually bind to an RNA that is actually negatively charged. Okay? So both are neutral at physiological pH to minimize toxicity. So as a result of the use of LNP, we have a fewer interaction with the anionic membrane of the blood cell when they are neutral at physiology pH and improve the biocompatibility for the mRNA delivery in vivo. So they are propagated in the acidic endosome after cellular uptake and thus interact with the anionic endosomal phospholipids to form a cone-shaped ion pair that facilitate membrane fusion and disruption. So we will escape the lysosome binding and release the cargo to the cytosol and finish its role, which is actually to being translated by the ribosome into a protein. So as a summary, so for mRNA vaccine, we will need to produce the final structure of the mature mRNA, which contains a 5' cap and untranslated region, start codon, coding region with pseudouridine, a stop codon, untranslated 3'N, as well as the poly A tail. So all of this will be starting from a plasmid construction followed by a in vitro transcription process. With that, I would like to thank your attention. And a last message for me before the Q&A session will be that our partner for NextGene, we do offer the GMP grade of the vector as well as the mRNA production. So if you need any further discussion on your specific experiment, okay, you can always contact us as well. Thank you for attention. And yeah, I'll pass this to Yiling and possibly answer some questions if you have any.
Thank you, Chi Hong, for the insightful presentation. We will proceed with the Q&A session. Feel free to drop your question at the chat box. Okay, so we have a question from in the chat box. So what is the advantage and disadvantage of capping during or after the transcription kit?
Okay, so during the transcription kit, the advantage will be quite obvious. It will become less laborious because you don't need to do an additional step. Okay, and then yeah, so that will be to me the main advantages of core translationing step because it makes things easier. And when we consider the post-transcriptional adding of the cap, it's basically if we want to achieve even higher capping efficiency. Okay? However, nowadays the core translational kit actually give a quite good efficiency that is up to more than 95% because last time before the advances of the technology, the kit might not be providing you such a good efficiency and therefore people choosing the post-transcriptional adding of the cap so that majority of the mRNA transcript actually obtain the capping.
Okay, so next question will be, for microbiome hosts, is it important to include capping in the target gene design?
This is for microbiome hosts, right? Okay, because the topic today is actually mRNA vaccine. So therefore, our host will be the eukaryotic cell. So therefore, we need the capping, the untranslated region, the poly-A tail. Okay, but for microbe, if we want to just express the protein, so we do not need the capping, the untranslated region, because as well as the poly-A tail, because the microbe, the natural mRNA does not have capping and those untranslated region. So those things actually in eukaryotic cell serve as a protection mechanism. So it will protect this particular mRNA from being degraded by the cellular RNAs. So if you are degraded, then the production of the protein will be low. So therefore, for microbe, in my opinion, you don't need all these things.
Okay, thank you, Chi Hong. So, and is there any question left from the audience? Feel free to just join the chat box.
Okay, so we have another question. It's about the use of the T7 polymerase. And okay, so my recommendation will definitely change the sequence. Okay, whether it will work or not, if I'm mistaken, I did read a small part from our partner NEB. It seems that it will actually greatly reduce the capping. So therefore, if you haven't started your experiment and if you are using clean cap, okay, we strongly suggest you to change the T7 promoter sequence to the AG. So perhaps I can share the particular explanation from NEB to Dr. Ching later.
Yeah. So there's another question. Is there any LNP formulation that is free to operate?
Okay. Thank you, Dr. King On, for this question. So unfortunately, I need to perhaps get help from NextGen partner about this particular question. So yeah, so we'll get back to Dr. King on later on this one about the free to operate LNP formulation.
Okay, thank you, Chi-Hong. So if there's no any question, that's the end of our webinar session. We hope you find today's session valuable. Yeah. So if you have any remaining question, feel free to reach out to NextGen product specialist, and feel free to just drop your inquiries and fill out the feedback form in the chat box. We do share the link there. Once again, thank you for attending. We are look forward to see you at our next webinar. Have a great day.
Thank you, everyone. Have a great day.